Michael, are you ready? My clock says 3:30, so we'll get started on time.
Sounds good.
All right, everybody online, thank you so much for joining us today for Hatfield's research seminar. My name is Cinnamon Moffett, and I'm the research program manager for Oregon State University's Hatfield Marine Science Center in Newport, Oregon, and I will be your host for today. Thank you so much for joining us. We are on a Zoom platform, so just letting you know that your mics, camera, and screen share have been turned off. We ask that you help us out by keeping it that way for the duration of today's event. But we do ask that you put in any questions you might have in the chat box at the bottom. If you lower your mouse down to the bottom of your Zoom screen, you'll see a little chat box and you'll be able to put in your questions there. I'll read those out to Michael at the end and we'll get to your questions as we get there. Also wanted to let you know this event is being recorded. So if you miss something or want to see it again, you're welcome to go to the recording. It'll be posted on the site I'm putting in the chat right now in just a few days. So you're welcome to go check those out. Also wanted to reach out to those students that are joining us today. If you're taking this for credit, Michael Banks, Professor Michael Banks is online and will be monitoring questions. So go ahead and ask away. One quick announcement for next week. Just wanted to promote next week's seminar, same place, same time. We have Christine Colloway and Katie Richardson from the NOAA Observer Program, who's going to be talking to us a little bit about the Northwest Fisheries Science Center Observer Program, what it is, how it works. So if you're interested in learning a little bit more about that, please join us next week. And with that, I actually have Bob Cowan is going to introduce today's speaker. So I'm going to hand it off to you, Bob.
Great, thanks, Cinnamon. Welcome, everyone. Very nice of you to come in from outside this beautiful day for this talk. I'm sure we're going to enjoy it very much. Our speaker, Dr. Michael Behrenfeld, is a professor at Oregon State University in the Department of Botany and Plant Pathology. He's one of our own, homegrown. He got his PhD at Oregon State in 1993, but he since then moved around gathering experience in a variety of different facilities. He went from Oregon State to working as a research scientist at the Brookhaven National Laboratory in Long Island, New York. And then he moved from there to be a assistant research professor at Rutgers University in New Jersey. In 1999, based on some of the work he's been doing, he accepted a civil servant position at NASA Goddard Space Flight Center before taking his current position back here at OSU in 2005. So following that route, his research has bridged a variety of fundamental understandings in the physiology and biochemistry with a broad scale understanding of biospheric functioning. And that's a way of saying he has a very large scale perspective of what's going on. And his work has been across many topics, including plankton ecology, remote sensing, development of satellite sensors, and optical approaches to ecology and physiological problems. All of this is application to assessing climate change impacts on marine plankton. So it's a big scale, big thinker, and I'm really excited to have him giving his talk today. He's going to be, title of it is Building Toward a New Lidar Era in Satellite Oceanography. Michael, thank you.
Thank you, Bob. Can you hear me okay?
Very well.
Good, okay. All right, so it's kind of challenge trying to think of what level to present at this talk, and I made an assumption that not everybody on the Zoom here knows as much about remote sensing as I do, and so I wanted to make sure I had everyone up to speed. The presentation is going to have some in-depth science stuff, but also just some stories about my involvement in satellite LiDAR oceanography. While the primary thrust of this talk is on satellite, it is actually on satellite LiDARs. I'm going to start out with a little bit of background on the more traditional ocean color remote sensing. So ocean color remote sensing is essentially the derivation of solar reflectance spectra from the ocean using measurements from the top of the atmosphere. And essentially what we're looking at is the color of water. And so on the pictures on the left here, you have, for example, at the bottom, the types of spectra on the left that would be associated with blue water with very little phytoplankton, and on the right, greener water with more phytoplankton, and how that shifts the spectrum of the light that comes out of the water. So what this technique is measuring is sunlight that has gone into the ocean or into a water body, interacted with particles or water itself, and then comes back out of the water and makes it up to space into a satellite sensor. The first satellite instrument that was built to measure or derive ocean color was called the Coastal Zone Color Scanner. And this is a picture of it here. This is really a proof of concept sensor. And the measurement bands that it had for oceans was very limited. It had 3 bands in the visible and 1 in the near infrared. And there's been many additional ocean color sensors since then, and they've added to the number of bands, but there's a lot of similarities in the basic approach. So essentially what these sensors are doing, excuse me, is they're measuring the top of atmosphere radiance as the satellite follows its orbit, which is typically a polar orbit. And the instruments either have multiple cameras looking across a wide area, or they use a rotating telescope to get this broad swath of data with each one of its orbits. And so the satellite will follow this same orbit over and over again. And as it goes around that orbit, the Earth turns underneath it. So each time it comes around on the daylight side of the Earth, it gathers a new piece of the planet in terms of observations. And one of the things you'll see in this graphic is that with every one of these orbits, it essentially misses a section in the low latitudes. The reason for that is that valid retrievals with these kinds of sensors are limited to the angle that they can actually retrieve from, so the solar angles they're dealing with. But the good thing is that every other day, the orbit is slightly adjusted from the previous day and it can fill in the gaps, okay? And so that's how it can get global coverage. This particular animation is for a new satellite that I've been working on a couple of years. So one of the things this animation is kind of misleading about is the coverage that you actually get. So one of the challenges with these traditional ocean color measurements is that they are unable to get valid retrievals through aerosols, clouds, even small holes in clouds. They don't get good retrievals where there's sun glint and when solar angles are steep. And so a typical day of measurements, this is a full day here, looks like this, and you can see there's a lot of missing data. But as you go from day to day to day, we start filling that in. So from 1 day to 3 days looks like this, to 8 days looks like that, and by the time you get to a month, you have pretty good coverage. Now you can take these months of data and years of data from multiple satellite sensors and start building a picture like this, where you can actually essentially see the planet breathing. Data have truly revolutionized our understanding of ocean biogeochemistry, carbon cycling. They've been used to greatly refine our estimates in net primary production in the ocean, to understand blooms and bloom phenomena, how the ocean responds to aerosol deposition. There have been thousands and thousands of papers written based upon these traditional ocean color measurements. While they are amazing contributors to our understanding of the planet, we've known that this technique has its limitations. We've known that from the very beginning. One of the challenges in ocean color remote sensing is that the actual signal that the satellite measures at the top of the atmosphere is dominated by light coming from the atmosphere and the surface reflection, and not from the oceans. So 90 to 95% of the signal is not an ocean signal, okay? And so we have to use additional wavelengths and models to try and remove the atmospheric signal, and that can have errors. The signal we get from the ocean color is also limited in terms of what depth it comes from. So basically the signal is coming from the upper 1/2 of an optical depth. And just for reference, the photic zone is 4.6 optical depths. So this is just, it's a number of meters below the surface. Because of this, it doesn't provide us any information about vertical distributions of ocean constituents. Another challenge is that the retrieved property, the water-leaving reflectance, is an optically integrated property. And what I mean by that is that it has contributions of scattering and absorption convolved into one signal. And we have to use models and assumptions to separate those signals. We don't have a direct way of separating them with the satellite measurement. As I mentioned, the retrievals are compromised by aerosol clouds, solar angle, glint, and other things. And this can be particularly problematic at high latitudes. And finally, because it relies on the sun, we obviously get no information about plankton systems at night. And it's because of these limitations of the technique that LIDAR, satellite LIDAR observations really can have a lot to offer for our new revolution in our understanding of aquatic ecosystems. So maybe most of you are familiar with LIDAR, but just in case, LIDAR means light detection, or is the acronym for light detection and ranging. And some of the strengths of the LIDAR approach are that the measured signal is coming from a known and very well characterized source, and that's the lasers on board. A satellite LIDAR makes its measurement at constant viewing geometry, so it doesn't have some of the complications of ocean color, which is looking at lots of different geometries in sun angle. Very minimal atmospheric correction issues, and with an advanced LIDAR, we can penetrate deep into the photic zone, far deeper than the half optical depth that ocean color tells us about. This will allow us to resolve vertical structure. Another thing that the LIDAR can do is it can penetrate through aerosols and thin clouds and still give us valid retrievals, and it sees well between clouds, so cloud gaps that would otherwise compromise ocean color. And of course, because it's bringing its own sun, its lasers, it can measure both day and night. Okay. So kind of my storyboard now for the rest of the talk is, well, how does it work? How does LIDAR work? Some notes from the field, a little bit about historical measurements of aquatic systems with LIDARs and how I got into this mess. And then going to space and a little bit of science, and then thinking forward, kind of some ideas of where I think we should be going with this. All right, so a little sip of water, or whatever it is I have in that cup. LIDAR 101. So basically, a LIDAR system is made of three primary components. It has a laser source, a receiving telescope that collects the signal from the laser when it comes back, and then that's sent to a set of detectors. And those detectors are measuring this signal roughly around the speed of light, okay? So the laser will send out a pulse, a very short pulse of light, and send it down toward Earth. And as that pulse of light goes through the atmosphere, it interacts with particles and molecules. And those interactions sometimes result in a backscattering of that light. And some small fraction of that backscattered light gets back into the telescope. And the same thing happens as that laser pulse goes into the ocean. When the detector measures the signal from that laser, it not only measures the intensity of the signal, but it also measures the time that the signal was made. And so you can create from this time and intensity data set a vertical profile of the structure in the atmosphere or the structure in the ocean, okay? This is an example of what a LIDAR curtain looks like. So what I mean by curtain is that unlike ocean color, one of the challenges with LIDAR is that it's basically only retrieving data at nadir underneath its flight path. So instead of providing swaths of data, it's providing a curtain of data, but it's a vertically resolved curtain. So this particular image is from a very advanced LIDAR system that was flown on an airplane during one of the campaigns I was involved in. And what you're looking at in the top picture is the LIDAR curtain as the plane flew from Chesapeake Bay to the shelf of Nova Scotia. And you're looking at aerosols resolved vertically and by type as the plane flew over this area. And the red smoke there was a distant biomass or wildfire from Canada. The bottom panel is the curtain of ocean retrievals for the region bounded by those two pink dotted lines, okay? And what you can see there as you go from the right over to the left is at first these retrievals were in very clear, relatively homogeneous waters. And then as you move to the left over toward the shelf, you start seeing subsurface features with the LiDAR. Then you get into the shelf bloom area where you have these really bright, I think I can use my cursor here, these red features. So right over here in the shelf, very high density of phytoplankton. And then finally moving over to the left, you see we start getting back into more mesotrophic systems with these subsurface particle maxima. So that's kind of what a curtain looks like. Okay, some notes from the field, all right? So airborne LiDAR measurements over aquatic systems have actually been done for quite a long time. The first one I am aware of was published in 1973 by Kim et al. And this was a LiDAR system that was trying to retrieve chlorophyll fluorescence from excitation at 532 nanometers. And it was just retrieving a bulk signal. And the instrument was strapped to the belly of a helicopter and flown 30 meters over the water to get these measurements. A little bit later in the 80s, the airborne LiDAR technologies continued to mature, and a Raman channel was added to help quantify the chlorophyll and phycoerythrin contents in the waters. And the example of some of those flights are shown in the right graphs. This is from a paper by Hogue in 1986. In the late 80s and early 90s, we started getting systems that were able to do profiling of the retrieved property, in this case, backscattering attenuation. And also in the early 90s, Frank Hogue, who I used to work with at Goddard, added a 355 nanometer channel and detector for also measuring the absorption of color dissolved organic matter. By the mid-1990s, there were some significant scientific applications of these technologies. One of them that I thought was interesting was by Jim Yoder, where he used a airborne LiDAR to retrieve chlorophyll information during the Jay Goff's North Atlantic bloom experiment. The idea here being that the airborne LiDAR could provide a lot better spatial coverage around the much more localized ship measurements. A year after that, John Martin used a LiDAR as well to look at the spatial distribution of phytoplankton chlorophyll as it responded to the first large-scale iron addition to the equatorial Pacific in an experiment called Iron X1. In the 1990s and up until today, another big player in airborne LiDAR measurements has been Jim Churnside at NOAA. And in some of his early work, he was using his airborne system to quantify and detect fish schools. Then he moved on to detecting plankton layers and zooplankton swarms, and then on to profiles of attenuation, backscatter, and chlorophyll. And actually, most recently, has been doing estimates of net primary production with an airborne LiDAR. Okay, now my story. So I really didn't have a whole lot of, not necessarily interest, but I wasn't really involved in the LiDAR story at all until I was working on a different problem. And that was that I wanted to try and develop an approach for quantifying phytoplankton biomass that wasn't chlorophyll, okay? Because chlorophyll has the very problematic issue of being an expression both of the concentration of phytoplankton and the physiology of phytoplankton. And so the idea I had here was we could use measurements of particulate scattering, whether it's backscattering or beam attenuation, to get an independent estimate of phytoplankton biomass, and then use that in conjunction with chlorophyll to look at physiology. So this turned out to be a pretty fruitful path, and it got me thinking of, well, how am I going to measure attenuation, beam attenuation from space? And that was a problem. And then I stumbled across a paper by Howard Gordon, which I thought was really interesting. And so this is a graph from his paper. And what it's showing is the relative field of view on the x-axis and the retrieved attenuation coefficient, okay? And the three different lines simply represent three different water bodies with different plankton populations. And as you go to the right over here, where you have a wide field of view, the attenuation that you measure is proportional to the diffuse attenuation coefficient. But what's interesting is that as you decrease your field of view over to the left, you see these curves start going up. And that's because as you get closer and closer to extremely narrow fields of view, the attenuation that you retrieve gets closer and closer to the beam attenuation, such that the intercept is the beam attenuation. So this made me think, well, I could make a LiDAR that could measure this attenuation at decreasing fields of view and get at my beam attenuation. So I put in a proposal, got funded, can't imagine that, and started working on building a LiDAR system for a ship. But the problem was that I became impatient. And I'm like looking at this and going, it's going to take me years to do this from a ship, years to do it on an airplane, and then maybe more years. I'm going to be retired before this goes to space. And so I started asking myself the question, are there any existing data sets that can indicate now that we can actually retrieve plankton properties from space? And I started talking to some of my colleagues at the NASA Goddard Space Flight Center, and it turned out there was. So skipping all the ship and airborne stuff, let's just go straight to space. It turned out that there was a mission on the space shuttle called the Lidar in Space Technology Experiment, or LIGHT, that took place in September of 1994. So the left-hand picture is a picture of light. It looks kind of like a garbage can pointing at the Earth. Okay, and then the bottom graph in the middle is a maneuver that they did. So the light instrument was a 3-wavelength neodymium YAG laser with a primary output at 1064 nanometers and then outputs at 532 and 355. It was a very powerful laser. We couldn't fly this from space anymore. It's not eye safe. But what it did, which was very interesting, is it did this 30-degree plus or minus maneuver over Lake Superior in the Gulf of California. The reason this is interesting is shown on the right, okay? So what you're looking at on the right is the top graph is the retrievals for the 532 nanometer channel through this 30-degree maneuver, and on the bottom, the 1064 nanometer retrieval. And you'll notice at the very top that there were 2 gain settings. So when the observational angle was greater than 15 degrees, they were using a high gain, and when it's closer to nadir, they're using a low gain. And in both panels, you see that in the low gain setting close to nadir, there's this big, bright red spot, okay? And the horizontal white line is the ocean surface. That's not plankton, okay? What you're looking at there is the reflection off the surface of the ocean being so bright that it blinded the detectors and they were ringing. And that's why it looks like you're still getting something at depth, okay? But off nadir, past 15 degrees, you see in the 1064 pattern at the bottom, there's no subsurface signal. But at the top there is. And when I saw this, I thought, oh my God, this is great. We actually can see the plankton from space. So after that, 2 important things happened, okay? The first was that NASA partnered with the French space agency CNES and built and launched a system called the Cloud Aerosol Lidar with orthogonal polarization, or Calliope. It went to space in April of 2006, and remarkably, it's still active and collecting beautiful data. It was definitely not made for doing the ocean. It wasn't made to do that. It was made to do clouds and aerosols. It did have a 2-wavelength system, and again, a neodymium YAG laser, the workhorse for lasers, 532 and 1064 nanometer outputs. It had 3 channels. The 532 had both a polarized and cross-polarized channel, which turned out to be important later. And it's a big system. So you can see the picture on the right. There's a guy standing next to the telescope. So this telescope has a meter diameter. It's big. And you can see the lasers there in kind of in the forefront. When the laser pulse makes it to the ocean surface, it's spread out so that the footprint is about 100 meters in diameter. One of the drawbacks for ocean studies is that the vertical resolution, because it was meant to study aerosols and clouds, is only 30 meters in the atmosphere and 23 meters in water. One of the nice things about it is it's a polar orbiting sensor with a precessing orbit, okay? And what that means is that it's not repeating the same orbit every single day. It's offset a little bit every single day. But over the course of 16 days, it comes back to a previous orbit. And what this gives us is full global coverage over a 16-day period. And that's what this picture is showing here. This picture at the bottom is showing the orbit tracks of the Calliope over a 16-day period. So it's giving us global sampling. Oops, go up, go down, try it again. Technical difficulties. The second important thing that happened is I met Young Hu. And Young is an absolutely brilliant scientist who works at NASA Langley. And Young and I met at an ocean color research team meeting, and I pulled him aside and I said, Young, I want to show you something from this space shuttle light experiment. So we talked about that, and I said, do you think there's any chance we could do a similar kind of retrieval with calliope? And so we started down that path, and Young used his brilliance and was able to, over the course of a few years, get an ocean retrieval. And so we presented this at the 2007 Ocean Color Research meeting for the first time. Kind of floored everybody. Here was a global retrieval of ocean particulate backscattering from a spaceborne LiDAR. And it shows all the basic features that we expect to see in terms of the distribution of phytoplankton. The next thing I wanted to do was actually go in and validate the retrievals. So we got some funding to put together a field campaign where we combined airborne LiDAR measurements, ship measurements of backscatter, and then flights with calliope to try and validate the LiDAR retrievals. And the findings of that study are shown here. So in the top panel, what you're looking at is a black line, which is the continuous underway particulate backscatter measurements from the ship. The red line is the calliope retrievals, as close to the ship as we could get them. And the green line is the MODIS backscatter retrieval from an inversion algorithm. And over most of that transect, you see that we have really excellent agreement between the ship measurements and the spaceborne measurements. The agreement's also very good when we compared the satellite LiDAR with the airborne LiDAR. So that's the bottom panel where the black is the airborne LiDAR and the red is the calliope. Again, the green is the inversion. So with that validation success, we put together a paper and published that in Geophysical Research, describing the first global distributions of plankton carbon stocks with an satellite LiDAR. Okay, so on to a little bit of science. So what do we do next? So one of the things I mentioned earlier is that ocean color measurements are very compromised at high latitudes because of cloudiness, solar angle. We get very, very little at all from ocean color from late August until early spring. And so what I'm showing in this graph is the left-hand circles are the ocean color retrievals for a given month. And the top circle on the left is the North Polar region, and the bottom circle is the South Polar region. And you can see there's very little ocean color data. The right-hand circles are the calliope retrievals for the same months. And you can see the calliope, because it can see through holes in the clouds, it can measure day and night, go through aerosols, is actually getting a lot more retrievals in these polar systems, right up to the ice edge, okay? So this was clearly an opportunity for us to get some new insights on the functioning of these high latitude systems during periods when we didn't have ocean color. So the first question we can ask with this data set, and the data set ran for a decade, is, well, let's first simply look at what are the long-term trends in polar zone integrated biomass. So looking only at the polar zone, integrated biomass for the whole region. And what we found is shown here. So I'll walk you through this one kind of slowly. So the two leftmost plots, I'll use my pointer over here, show the long-term record of ice-free area in the top for the North Polar Zone and the bottom for the South Polar Zone. The gray shaded area is the calliope era, okay? And the white on the left, there is no calliope data. And so what we see in the North Polar Zone, of course, is that there was a long-term increasing trend of ice-free area in the North Pole, and that's received a lot of press. Interestingly, during the calliope area, the general trend kind of flattened out. In the South Pole, it's different, it's kind of the opposite. The trend in ice-free area there was relatively flat until the calliope era. And then we see this downward trend. So let's ask the question, well, what happened with the plankton? Okay, so the right-hand graphs, now the polar north is the middle column and the polar south is the right-hand column. Okay, in the top and bottom plots, the black line is the same, and that black line corresponds to the polar zone integrated biomass, okay? So for the north and the middle and the south and the right. And then the top panels are showing that change in biomass, or those changes in biomass compared to ice-free area. And so we see something very different between the two polar systems. In the North Polar System, the variations in biomass don't seem to be tracking the ice-free area very well at all. But in the South Polar Zone, they are. On the bottom panels, what you're seeing is the relationship between polar zone integrated biomass and the average concentration of phytoplankton per cubic meter. So what this is showing is that in the North Polar Zone, really what's happening there is the variations during the calliope area in terms of total polar phytoplankton biomass is really being dictated by the small scale ecosystem processes that are regulating the concentrations. Whereas in the South Polar Zone, that relationship is nowhere near as strong, and ice-free area really is driving that system for this era. So what else can we do with this data? Well, we can now ask, how does having a truly complete annual cycle coverage with calliope provide insights on the dynamics of polar system blooms? Okay, and I'll walk you through this one too. This is a little more complicated. So think about an annual cycle in phytoplankton biomass. It's going to have a bloom, it's going to crash, it's going to come down and have winter minima. So you can take that concentration data and derive the rate between each time point, in this case monthly, the rate at which it was changing. Okay, so that's what I'm plotting in this graph. So this is the biomass rate of change. If biomass is not changing, the rate is obviously 0. That's the horizontal dashed line. If it's decreasing, the numbers are negative. If it's increasing, the numbers are positive. So basically what you have here is you have decreasing biomass from January and early spring. Here's your blooming period, and then here's your declining phase again. Same thing with the South Polar Zone, but I've just switched the axes by 6 months. Okay, what drives this? What's driving this annual cycle? Well, we can compare this biomass rate of change to the actual division rate of the phytoplankton, okay? And when we do this, we find something very interesting. So the initiation of the blooming period up here is when the values cross from negative to positive. When it crosses over, that's the initiation. And we find that initiation, if you follow this purple dashed line down, happens when the division rates are very low. Basically, as soon as the sun starts coming up in this region, the bloom gets initiated. The next thing that's interesting is the fastest rate of biomass accumulation is happening at only an intermediate rate of division. The crash of the bloom, which is where it goes from positive to negative, and this green line, happens when the division rates are maximum. And then finally, the maximum declining rate happens when the division rates are about the same as they were when it was at its maximum increasing rate. Okay, kind of interesting. Same thing is for the South Polar region. So why is this? Well, if you look at this like I do, stare at it for a while and think about this, you can kind of visually look at the blue lines down here and intuitively derive or create the derivative of that blue line. And when you calculate the derivative of the blue line, you come up with this pattern. So here I'm showing the same gray symbols or open white symbols, biomass rate of change. And the red symbols now are the rate of change in division rate. So the biomass rate of change in units per day is being driven by a property in units of day to the -2. And why is that? The basic conclusion from these observations, and this was the first truly clear demonstration of this, is that the annual cycle in biomass is driven not by the absolute division rate, but by accelerations and decelerations in phytoplankton division rates. And this is not because of the bottom-up processes per se, but because division and loss rates are perpetually tightly coupled in plankton ecosystems. But there's a short temporal lag between a change in the division rate of the phytoplankton and a change in the loss rate due to predation by zooplankton, viruses, and other things. So this is the basis of a hypothesis called the disturbance recovery hypothesis. And it provides predictions about how blooms will change in the future. Oh wait, totally switching gears. All right, so that was the polar thing. And then I got interested in something called the DVM, the diel vertical migration. It's the largest animal migration on Earth. It happens all over the global oceans, happens every night, okay? And it's a migration of animals of many different sizes and trophic levels. And this story started out rather sleepily. So I was on a cruise in the North Atlantic, and it was the middle of the night, and we were doing one of our standard CTD casts. And as the CTD was going down, I was watching the screen, and chlorophyll was uniform in the mixed layer and decreased below, kind of like we expected and kind of like we saw every single night. But what was interesting this time is that as the CTD came up a short time later, in the mixed layer, which should be uniform, there's this huge wedge of phytoplankton that's gone. I was like, wow, look at that. And Pete, who's the big guy here sleeping by his computer, was simultaneously measuring acoustics. And Pete was looking at his data and said, Mike, that corresponds to this mass influx of vertically migrating animals. They came through and just wiped out the phytoplankton. I was like, wow, that's a bigger effect than I ever envisioned for this DVM thing. And so I started getting interested in DVMs. And then the next thing that happened, I was at an ocean sciences meeting, and this young guy named William Burt came up to me and said, Mike, I got something I want to show you. So I walked over to his poster, and he showed me this time record, and it's really interesting. So what this is showing is time on the x-axis. Black bars are night, white bars are day, and particulate backscattering is on the y-axis, okay? And you can see every night, or roughly at night, there's all of a sudden this big bunch of spikes in his data. And I was like, I know what that is. Those are diel vertical animals coming into the surface layer and getting seen by your sensor. And the reason they show up as big spikes is because they're fairly large critters compared to phytoplankton, so they're bright targets. But they're very, very much less abundant than the phytoplankton, which is the purple line down at the bottom. But what's important is that when you integrate the area of the spikes, it's roughly half of the total nighttime signal. And so this made me think, well, maybe we can see this from space. Can we see a day-night difference in the satellite LiDAR retrieval because of these vertically migrating animals? So that was the next harebrained idea I went chasing after. And after a lot of work, we came up with this fundamental finding, okay? So what I'm showing here is the day-night difference in the satellite LiDAR retrieval of particulate backscatter, so delta BBP, which is simply calculated as the BBP measured at night minus the BBP during the day divided by the BBP during the day. So it's a normalized value. And what you see is very clear regional patterns in the tropics and subtropics. And these correspond to, obviously, features in the distribution of phytoplankton. You'll also see that the scale on this goes from -8, that should have been a percent, I think. Yeah, it should have been a percent, -8 to +8. And so the question is, well, why are there negatives, right? If this is a diel vertical migration, why are there negatives? It should all be positive. Well, the answer to that question is that delta BBP is not just a DVM signal. It has two fundamental components. It has a phytoplankton component and it has a DVM component, okay? So we need to tease those apart. So the phytoplankton component looks something like this. Okay, so this is a mean cycle that was measured during non-bloom conditions in the Mediterranean. There's very, very little diel BBP data from the field. This is one we used. Okay, and what you see here is that right around dawn, the backscattering from phytoplankton starts to increase and comes to a peak mid to late day. And then it decreases. And what primarily drives this cycle is changes in the carbon concentration of the phytoplankton. They're photosynthesizing during the day and respiring that carbon during the night. And that changes the refractive index of the phytoplankton. So the phytoplankton cycle alone, what we expect is that the peak, which in this graph occurs at the daytime overpass time of the Calliope LiDAR, is going to be, the daytime peak is going to be higher than the nighttime peak. And so if you take my night-day difference, you're going to end up with a negative, okay? Okay, so in the absence of DVM animals, the night-day difference is expected to be negative due to the phytoplankton. And then as the nighttime abundance of DVM animals increases, you're going to have a rise in that nighttime signal to make the number either less negative or even positive if there's a lot of DVM signal. We also know that the magnitude of the phytoplankton cycle is dependent upon some environmental factors, such as their division rate and photoperiod, and also species composition. So when we factor out using a model, the phytoplankton element of that cycle, and then compare the residual DVM signal to field measurements from net tows of DVM animals, we find these results here. So what you're looking at is on the left top is an annual average cycle of DVM biomass for the Hawaii Ocean time series, which is the leftmost star on the bottom graph. And the same thing for the Bermuda Atlantic time series on the right, and the rightmost star on the bottom graph. So what we see is that similar annual cycles are apparent in these two records, noting that these are not coincident records in time, okay? But the general patterns are pretty reasonable. And these HOT and BATS data also provided us a first order index on how to relate the DVM signal from space in units of BVP to an actual biomass of DVM animals. So then what we did is we wanted to ask another question. Okay, so if we can do that, and we have this 10-year record, do we see regional trends in the DVM signal that we're getting from space? Because we certainly can't do this from ships or net tows. They just don't give you the coverage that a satellite sensor does. So what I did is I broke up the subtropical area into these colored zones, which are have little labels on them. And I looked at the trends in DVM signal over the 10-year Calliope record for bins that stayed within their given boundaries, okay? And the result of that analysis is shown here. It's intriguing, and I don't know what it means. Okay, so what I'm showing here are the temporal trends, the decadal temporal trends in percentages in colored boxes, and each box represents a 6 degree by 6 degree bin, okay? If the trend is significant with a p-value of less than 0.05, I put a little black dot in it, okay? So you can see the trends. The red areas are increasing DVM signals. The blue areas are decreasing. White, of course, means very little change at all. And then I asked the question, okay, we see significant trends in a lot of places. The direction's different in different regions. How is it related to the phytoplankton production? And so I did another analysis, and I said, how well is the DVM trend linked to trends in net primary production for that bin? And if the relationship was positive and significant at P less than 0.05, I outlined the box in a solid black. And if it was significant but inversely related to NPP, I outlined it in green. So what we see in this is that, in general, the trends tend to be positively linked to production of the phytoplankton. But there are regions, particularly on the west side of Africa, where the dominant relationship seems to be negative. I have no idea why. This is just an open question, and it's cool, right? But it's just kind of a first glimpse at how we might think about using satellite lidars to study something other than phytoplanktons, you know, higher trophic levels, and say something about what's going on with them. And it's important because the DVM is a major conduit of carbon from the surface of the ocean into the mesopelagic, many, many millions of times. Okay, so that's just kind of a little bit of science. Thinking forward, what next? Where do we go? So first of all, we need to think about Calliope as not satellite lidar oceanography. It's a proof of concept. It's the same thing as CZCS. It's just the proof of concept. But it's very important because Calliope has fortuitously circumvented what I call the graveyard of good ideas. And there's lots of people at NASA centers that are thinking about all kinds of cool satellite missions, but going from the field to space is the graveyard. Calliope gave us a chance to get there without building an instrument to do it. And it has enabled some neat new science. But the question now is, what could we do if we actually built a satellite lidar that's optimized for ocean research? Calliope is what I call a simple elastic backscatter lidar, and it presents an ill-posed problem. It provides one measurement retrieval, and that's called the attenuated backscatter. and it's composed of two unknowns, the backscatter coefficient and the diffuse attenuation coefficient. In order to get backscatter out of that, we have to bring in ancillary data and or biooptical assumptions, and that has potential errors. How do we solve this? There's another LiDAR technique that's called the high spectral resolution LiDAR, HSRL. And I showed you data from an HSRL early on when I showed you those first curtains. That was HSRL data. So when you shoot a laser into the water, what happens is that some of it will interact with particles and get backscattered, and it will be backscattered at the native wavelength of the laser. But it can also interact with water molecules, and the water backscatters it at a slightly shifted wavelength. It's called Brion scattering, okay? The technique of the HSRL is that it adds an additional detector. So I'm showing kind of a schematic over here. So over here on the left is my LiDAR system, okay? Here's my laser source. And what I've done is I've tuned this laser to the very narrow absorption line of an iodine cell. Okay, so now I'm shooting this LiDAR down here. It comes back with a signal from the ocean, goes into the telescope, and that signal is split off into our two standard cross and copolarized channels, and then a third channel, this iodine cell. And the light that goes through that iodine cell hits this one here, and all that's left in the signal is the Brion scattering, not the combined scattering of the Brion and the particulate, okay? So what we've enabled us to do is that water backscatter is very well known. And so we can get the attenuation coefficient from the Brion, the iodine channel, and then use that to derive the backscatter directly from the other two channels. So that solves, we have a well-posed problem. We have two measurements and two unknowns. And this kind of system has been flown many times now over the ocean in an aircraft situation. I'm just showing some of the flights that we've done with HSRL, but there's many more in many other ocean regions. And the bottom right is kind of interesting. This is the particulate backscatter coefficient retrieved from the HSRL airborne LiDAR on the y-axis and coincident in situ BV measurements from a ship. And what's interesting about this is I want you to note the coloring. So the different colors correspond to different depths of the retrieval. And you're seeing that we're getting good retrievals down to 3 optical depths. So that's pretty far down into the photic zone. It's very exciting. So what would a LiDAR mission look like if we actually designed it for the ocean? This is where I'm hoping to go in the future. First of all, above and beyond the capabilities of calliope, we want to add HSRL technology, okay? We want to increase the vertical resolution to meter scale resolution so we can start retrieving profiles of the plankton. With respect to the laser, we kind of have two options. We have the workhorse neodymium YAG laser with its 3 wavelengths, but there are also new technologies being developed for a blue laser, and particularly a blue laser around 480 nanometers. And this is beneficial because that wavelength corresponds to one of the solar Fraunhofer lines. So that decreases our background noise and improves our signal. And it's very likely that with that kind of a LiDAR, we could get down to the very bottom of the photic zone. We want to also think about adding some additional bands, Raman bands, a chlorophyll fluorescence band, things like that. So there's a lot of possibilities here in the future.
Mike, can you try to advance your slide 1 forward? I think we're stuck.
Uh-oh. Where are you at? What are you seeing?
We're seeing the HSRL slide, the high spectrum, the one right before the one I think you're trying to show.
Okay. What are you seeing now?
Same slide.
Okay.
So I think that's slide 41 that we're stuck on.
Maybe you could close off your video, Michael.
Okay. How do I close off my video? Stop video.
Yeah.
There we go. Stop video.
Huh. Still see you.
Are you still there?
Yeah, I'm still here. Can you see me?
We can't, well, we can see a frozen version of you and we still see slide 41.
Okay, well, it just gave me a note that the program has crashed and I'm going to have to start again.
Okay, so do you want to stay on the line and I will share my screen and we can, what do we have, 2 more slides?
We have 2 more slides, yeah.
Okay, so everybody hang in there. Give us just a second. Oh, we've got it.
You got it.
We've
Great.
got it.
Okay, so good. And I
So
will,
we see the black slide that says, what would a LiDAR mission look like if?
okay. So what I've talked through is the top bullets, okay, the HSRL approach, the meter scale, the different laser options and different types of bands we could add. And it's not just pie in the sky. There actually is a mission that NASA is planning for right now called the ACCP mission. It's an aerosol cloud condensation precipitation mission. And it's intended to have a LiDAR. And one of the technologies that is on there is an HSRL. So we don't know if it's going to happen. There are other competing concepts, but there is a hope for an HSRL there. The Chinese Space Agency is rapidly moving towards developing a blue LiDAR mission for the oceans. So we'll see what happens there. It's a challenging thing to do. The other thing that's going on is that blue lasers are being developed for satellite applications that have very low, what we call wall plug power requirements. And so they don't require as much power as our standard workhorse neodymium YAG. And this might allow us to do a blue laser ocean mission on a small sat, which is something we could compete for in some of the open competitions rather than directed missions. Okay. Looks like my screen is now stuck. Can you see what do you see now?
We got the thank you slide.
All right, that's it. Thanks everyone for sticking with me. I hope it was of some interest. It's fun. And I'm happy to answer any questions.
Wonderful. Well, we almost made it all the way through without technical issues. So we're going to get some questions. Let's see. Let me work through these just a second.
And I would have been on time if we didn't have technical difficulties.
That's true. All right. Our first question that's coming in says, although it might not be optimized for research, is LiDAR data from the most recent satellites, such as ISAT launched in 2003 or ISAT-2 launched in 2018, applicable for measuring biomass and plankton in the same way as Calliope?
Yes, it is. Yes, it is. Kelsey Bisson in my group is working on a newly funded project to use ISAT data to retrieve plankton properties at higher latitudes. And Yang Hu and his group at Langley have actually retrieved some under ice plankton populations with the ISAT, as well as ice edge plankton populations with vertical resolution, unlike the Calliope. So yes, absolutely.
Nice. So there's another question here that's asking about the DVM. Is it similar for different plankton from place to place? Like, can you kind of tease that apart?
It is different. It is different. So the different types of animals that are migrating vary from one area to the other. And one of the big challenges, one of the big opportunities that we have right now is to try to understand which animals are actually responsible for the signal and how that varies from region to region. So in our first paper on this, we did a back of the envelope conversion to biomass, but that's all it is a back of the envelope, right? We're using two field data sets and making a conversion. The expectation is that there are going to be specific elements of the DVM that actually have a significant backscatter signal while others do not. Most likely that the largest part of that is going to be the migrating zooplankton. The fishes and other things which are much less common are going to be a smaller element to it, even though they're brighter targets. So one of our challenges is now to try to understand the details of this signal and do better estimates of relating it to the biomass of the migration.
So this next question kind of builds on that a little bit and is asking you if you could elaborate a little bit more on using LiDAR to capture the fish message, like Jim Kirsten's work,
Sure.
Since fish might go deeper than the LiDAR can detect.
Yeah, so the detection that we're doing for the DVM, remember, is just nighttime, and it's just the first 23 meter bin. So there's a lot of rich dynamics in the DVM in terms of how far they get up to the surface. You know, if they are coming up and they encounter food before they get to that surface bin, we won't see them, right? They have to actually get up to the surface bin of 22 meters in order for us to see them. So another challenge that we have, another opportunity that we have, is try to understand what environmental factors drive that migration in terms of its vertical distribution, And how do we relate a signal that we see in the upper bin to something below? Now, the question is easier answered if we have a blue LiDAR in space that can actually give us the vertical profile of that DVM signal. Then we could say really something about it globally. One avenue to pursue it at the moment is to start looking at, historical ADCP measurements from acoustics measurements from ships and see what we can learn about the environmental drivers of the migration behavior from that. But until we have something that can penetrate deeply from space, we're kind of challenged by trying to relate the surface signal to the full DVM.
Another question is, so is there a known anticipated expiration date for Calliope?
It was built to last for 3 years. And it's now flying. When did it get launched? 2006. We're 14 years into it. About 2 years ago, I was talking with Chris Hostetler at Langley, and he said, "Mike, I'm afraid it's lights out for Calliope." They thought they were going to lose their second laser, and it didn't. So they were able to keep it going. So the biggest challenge with the LiDAR systems historically has been that the lasers burn out. The first ICE mission with a LiDAR didn't last hardly at all. And just the lasers burned out, a mechanical mistake in the welding. Calliope, fortunately, has just been going and going and going and going. So it's going to end soon. It really will. I'm sorry. I don't want it to, but it will.
I have another question here that's looking at actually going kind of back towards a smaller, I'll just read the question. Would there be value in flying LiDAR and nearshore drones for getting more real-time localized data of this type, kind of thinking going backwards from satellite, but maybe more accessible?
Absolutely. I'd like to think about, kind of with my NASA hat, of thinking about our NASA satellite observations as being part of an Earth system, part of a system observing system. In other words, that satellites don't answer all the questions, airborne don't answer all the questions, autonomous sensors don't either, but the combination of them would be a phenomenal thing. Another piece that I've been really trying to push is that if we do move toward a satellite LiDAR for oceans, we should think very seriously about, deploying a global fleet of BioGeo-Argo profiling floats that can get below the LiDAR, can validate the structure we're seeing. The airborne stuff with the LiDAR is still going on hot and heavy. We've got lots of really great data in more regional and coastal areas. The coverage you can get with an airborne in terms of within a region is better than the orbit that repeats every 16 days. So yeah, absolutely. We should think about an observing system, not just a satellite.
Nice. So wondering if the delta of the DVM versus the delta of primary production is variable.
Yes.
Thank you.
Yes, it is. So if you look at the simple question that I asked in that graph was, are the trends correlated? Not are the slopes of the lines all the same? And the answer to that question is no, they're not. There's variability in terms of the strength of that DVM signal and the magnitude of the change in primary production. So no, it's not the same everywhere.
So Francis, I butchered your question. So if that didn't answer it, go ahead and type in an expansion to that. All right. It looks like questions are slowing down here and we're getting some thank yous, and that was wonderful and amazing work. Very cool. So if anybody has any last questions, go ahead and type it in. Otherwise, I think I have somebody saying I skipped a question. Hang on, they're coming in too fast for me to read. So Francis, let me see. Hang on just a second. I'm gonna. Francis, I just unmuted you. Do you want to ask your question?
Yeah, that's super cool, Mike. I just wondering that the phasing and anti-phasing of predator versus prey change. If you're looking at basically Lotka-Volterra cycles, one that's maybe it's a quarter cycle, one's a half cycle, so they're phase or interface, which is of course fundamental stuff in the field.
So is it behaving like that? The coupling that we're seeing in the plankton is on the order of days to maybe a week. It's very, very tight. So it's not quite the longer term coupling between predators and prey in classic Locke-Volterra relationships, but it is similar in a sense. Yeah, it's a very different. I think that one of the things that I'm excited about with this interpretation of blooms is that it's very much in contrast to our traditional thoughts on critical depth hypotheses and bottom-up controls, in that really the dynamics of blooming are largely an expression of ecosystem relationships that are related to environmental variability in bottom-up factors. But the core of it is really the ecosystem, the trophic dynamics that's driving it. And it's very commonly thought that the dynamics of biomass are proportional to division rate, when in fact they're not. They're proportional to the rate of change in division rate, which can be very independent of the division rate itself. Did that answer your question?
Yeah, I'll follow up a little bit because I think this is super cool.
Thanks. I've got a lot of papers on it.
Good. Another question that's coming in is, will the methods that you talked about work in inshore waters such as in estuaries?
The technological methods?
Mm-hmm.
The technology, yes, it can work in coastal areas, bays and things like that. But it depends on what you're trying to get out of it. So in the open ocean, the dominant backscattering particles are living organisms. And in more coastal turbid areas, a large portion of the backscattering will be coming from suspended sediments. And so taking that backscatter signal, or the attenuated backscatter signal, and converting it to biomass is going to be a challenge in that sense. And of course, you're also going to have a challenge with penetration. So it depends on how much depth you want. The HSRL Lidar has been flown over the Chesapeake quite a few times, and they do get retrievals. But it depends on what property you're really trying to get at. If you want to get phytoplankton biomass, it's going to be challenging because of the suspended sediments and other non-living detrital suspended particles that are going to be influencing strongly the signal you get. Does that make sense?
Ted, if that didn't answer your question, go ahead and type in the next one, and I'll try to expand on that. Victoria, do you want to ask your question? I've made it so that you can unmute yourself if you'd like.
Oh, thanks. I was just wondering if you could elaborate on how LiDAR was used by Jim Cherson to work with detecting fish population size, and how that could translate to other marine megafauna like juvenile sea turtles or something like that, where they tend to stay higher up in the water column. Yeah,
Jim Chernside's stuff is, to be honest with you, it's basically the same retrieval that we're using to get the phytoplankton. He's looking at the backscattering signal that he's getting from his LiDAR, but he's defining fish based upon the strength of that signal. So if you hit a school of fish with a LiDAR, it's a big signal, much bigger than the phytoplankton, and not as spatially uniform. And so what he's really looking for there are essentially anomalies from the background, a big anomaly from the background, and using that to get his fish. So, big animals, I had mentioned in the DVM, when we were talking about the DVM, that most likely the signal we're detecting from space is zooplankton and smaller animals, and probably not the larger ones. But when you get concentrations of larger animals, you'll get a big signal.
Did that answer your question, Victoria?
Yes, thank you very much.
All right, and we have pushing time just a little bit, and it looks like, Michael, you might have frozen again. So for those folks that are still online, thank you so very much. If you have additional questions for Mike, reach out to him directly. Otherwise, I hope to see you next week, same time, same place. So thanks everybody for joining us.
Thank you, Cinnamon. Thank you everyone for listening. I hope it was all right.
It was great. Thank you so much. We can still hear you, so that's great. I was afraid we lost you all together.
All right.
So much.
Bye-bye.
Thank you.
Bye.
Thanks, everyone.